Hello and welcome. My name is Marva Ward. I'm the National Product Educator for the Now brand of supplements in Canada. And today I'm going to be speaking about epigenetics, nature versus nurture. Epigenetics is a really exciting discovery within genetics. And so what we'll do is first we'll start talking about genetics on a very basic level. Gene is a segment of DNA, and it's passed down from the parents to the children, and it confers a trait of the offspring. Genes are organized in packaged units called chromosomes, and histone is the chief protein component of chromatin and DNA. It acts like a spool around with which DNA winds, and I think with a pictorial on slide number 3, you'll see you have the genes, the DNA, and really in a chromosomal structure, and ultimately is within the cell and is part of the genome. Another way to look at it too, again, is the genes within the nucleus of the cell. Looking at it at a closer level, the chromosome, and then the chromosomes are made up of DNA and genes. I think if we look at the right hand pictorial on the bottom, you will see the histones which are acting like the spools with which DNA is wrapped around. And ultimately, that is what chromosomes are made up of. Humans have 23 pairs of chromosomes. 23 pairs equal the human genome, and the human genome consists of 3 billion pairs, base pairs, that continually replicate, and almost like photocopying. However, sometimes there's a variation in a single base pair, and this is called a single nucleotide polymorphism, or a SNP, where this plays a key role specifically for two types of SNPs is MTHFR. Now, the MTHFR gene there's two genes, an A1298C and a C677T. If you inherit this variation of the MTHFR gene, the ability to activate folic acid is compromised, and to produce the MTHFR enzyme is compromised as well. The Mayo and Kant variation play a role in neurotransmitting activity, our ability to degrade catecholamines, etc. But for this particular presentation, we're gonna focus on the MTHFR. So epigenetics is on top of genetics. That's literally what it means. Additional information that's layered on top of DNA. It refers to changes in genetic material that invoke modification or mutation, not necessarily related to historical gene replication. So the genotype, which is a set of genes responsible for a particular trait, plus the environment, equals the phenotype, which is the physical expression of that genotype. It refers to heritable changes in gene expression that does not involve changes in underlying DNA. A change to the phenotype without a change to the genotype. A perfect example of this would be twins, identical twins, where you have one twin living its life in a completely different way than the other, and ultimately, when they reach perhaps the age of 50, one of them may be profoundly ill with many different types of so-called inheritable diseases, whereas the other one is in perfect health simply because of the environment that they've lived in and the lifestyle that they've lived. Epigenetic mechanisms. There's several of them. Histone modification, methylation, acetylation, and particularly what we're going to talk about here today is methylation. over and under, causing inappropriate gene expression. DNA methylation, too much or too little, leads to unwanted alterations in the cell. Again, the environment having an effect on the structure, expressing itself in a particular physical manner. There's small and non-coding RNA and chromatin architecture. But as I say, what we are going to look at as far as epigenetics is concerned is the methylation aspect. Diseases that are influenced by epigenetic biochemistry, alterations in the epigenetic pathways, have shown to be implicated in modern disease, particularly when we're talking about methylation. Cancer, hypo and hypermethylation. I'll speak a bit about that as we carry on. Cardiovascular disease, neurological disorders, aging. Poor methylation is said to be, aging is said to be the result of maybe inappropriate methylation. The faster we age, perhaps the more we're experiencing inappropriate methylation. Diabetes type 2, obesity and infertility. Epigenetic modification and methylation, these mechanisms, in addition to other transcriptional regulatory events, DNA to RNA, regulate gene activity and expression in response to environmental stimuli. Inappropriate or inadequate variations affect genetic modification. So hypo and hypermethylation or acetylation, underlying causes linked to the onset of many diseases. Hypomethylation, oncogene, it's a potential to cause cancer. Hypomethylation is overactivity of methylating. The activation of chromosomal instability is what it produces. And we'll speak a little bit about that. But hypermethylation is associated with silencing of tumor suppressor genes. And this is really where, again, the big focus will be on nutrients that will regulate methylation. The significance of appropriate methylation and homeostasis is profound. Detoxification in the liver, brain function, neurotransmitting synthesis and utilization of dopamine and serotonin, protein synthesis, genetic transcription, preservation of telomeres, chromosomal and DNA tails, which we've heard a lot about in our industry over the past maybe 6 years, where the preservation of telomeres is a surefire way to, number one, slow down the aging process and perhaps reverse disease. Cardiovascular disease, the reduction in homocysteine levels, or is what you're looking for. Cardiovascular disease is often related to too high of homocysteine levels, which is a direct result of poor methylation. So why are we seeing faulty methylation? What is going on? What is the environmental stimuli that's creating this problem? Known or suspected modification stimuli would be stress, absolutely. Heavy metals, pesticides, diesel exhaust. As we look at this list, we're looking at the culprits that are typically known to create many diseases. And so does it start with faulty methylation? That's something that certainly science is looking at. The aromatic hydrocarbons that are found in fossil fuel, hormone regulation or lack of it or too much of it, radioactivity that we're exposed to, viruses, bacteria, and absolutely poor nutrition. This is where things become exciting, because if we can control it simply through nutrition, which it appears to be more and more what we can do, then this is a pretty exciting finding, that it's a matter of looking at our nutrition and regulating nutrition to be able to slow down or stop the expression of certain genes. Methylation is carried out by specific enzymes, each one using specific nutrients. And folic acid, B vitamins, SAMe, any methyl donor, they're key components of the methyl-making pathway. Diets high in these methyl-donating nutrients can rapidly alter gene expression, especially during early development when the epigenome is first being established. Where they really discovered this to be true was the agouti mouse. The agouti mouse is a very obese, yellow, unhealthy mouse that develops cardiovascular disease, type 2 diabetes, and it's all based on their food, the sort of the mouse chow that they're eating. It was interesting to note that generation after generation of the agouti mouse has the yellow fur, very obese, ends up dying young, and with cardiovascular disease and diabetes. Lo and behold, in one generation, with a pregnant agouti mouse, they started feeding the mouse folic acid, choline, any type of methyl donors that might help, and they bore babies that were brown, muscular, lived long lives, and prospered, and their generation did the same. So I think it's interesting to note here a few things, that gene expression was reversed in one generation simply by altering their food choice and adding methyl donors to the mouse chow. This is a simplified methylation pathway, although tongue-in-cheek. It's not simplified whatsoever. It's very complex. The next slide. So this is definitely more simplified as far as showing the methionine pathway. The amino acid methionine converts to SAMe, donating a methyl donor converting to SAMe with other nutrients like folic acid that are cofactors in this production. SAMe converts to homocysteine. And this is where the cycle seems to stall, and this is where a lot of the problems start to occur. The MTHFR enzyme is compromised in some cases, definitely because of the MTHFR gene. And ultimately, most people are low in B12 and B6 and folic acid, whether they have the MTHFR gene or not. And ultimately, this is where things start to get complicated. Homocysteine levels rise, and other medical problems start to occur. Methylation pathway, here's another more descriptive pictorial of it as well. Again, methionine to SAMe, to homocysteine, and then ultimately back to methionine. And ultimately from homocysteine back to methionine, you require the B12, B6, and folic acid. When consumed food folates, predominantly methylfolates that we're consuming, are hydrolyzed into monoglutamate form, which is folic acid. So we may take folates in, but it is changed to folic acid initially in the body. It occurs in the gut prior to the absorption of the active transport across the intestinal mucosa. So before entering the bloodstream, the monoglutamate form, the folic acid form, is reduced to the tetrahydrofolate, or the THF form. And it's converted to either a methyl form or a formal form. Main form of folate in plasma is 5-methyl-THF, or MTHF. Here's the folic acid cycle, showing you again at the beginning. Dietary folate converts to folic acid, and ultimately folic acid converts to the methyl form of folate, THF. Folic acid is reduced within the cell to its metabolically active form, THF, from 5,10 methyl tetrahydrofolate, which is reduced again to MTHF. This is the form of folic acid that's used in the body for normal cell division, numerous other fundamental processes such as the synthesis of neurotransmitters and DNA, as well as completing the methylation pathway. So the MTHFR enzyme, it's needed to convert THF to the methyl form MTHF as far as folic acid is concerned. So it is the enzyme MTHFR, or methylene tetrahydrofolate reductase, that is used to convert folic acid to the methyl form of folic acid. And of course, this is used to detoxify homocysteine and to convert back to methionine to complete the methionine pathway. Saying it in another way, folic acid, MTHFR gene, and enzyme. It's the MTHFR gene that produces the MTHFR enzyme. And it is the MTHFR gene that is often a SNP, or it is some way altered. So the methylene tetrahydrofolate reductase converts our folic acid into a methyl form so that we can then convert homocysteine back to methionine, the completion of the methionine cycle. So as I mentioned earlier, the MTHFR mutation, or the SNP, is most common MTHFR gene mutations, and they're found in specific positions, 677 or 1298, which I had mentioned as well. The reduction of the production of MTHFR enzymes results in the increased homocysteine levels and continual lack of completion of this methionine pathway. The epigenetic principles applying ultimately result in disease. So giving you an example of what was studied, how they showed the reduction of the enzyme with these two mutations. Now, the genotype can have two different mutations, and if you'll see that if the two are normal, there's 100% enzymatic activity. Now, if the 6T7CT in one individual with a normal in the other, there's a 66% enzyme activity reduction, or there's a 66% enzyme activity. And as you can see, the next slot over with this 677TT variant shows a 25% enzyme activity, etc., etc. You can follow this this chart to show you the reduction. Having both the 1298 and the 677 is an 83% enzymatic activity, and again a 48 enzymatic activity. So the reduction of the activity of the enzyme is based on whether there's one gene that's been affected, or both parents, or one parent, or both genes. I think what's interesting is that studies show that supplemental folic acid and the maternal MTHFR deficiency has reported that reduced activity under low and normal folate levels, yes, but normal activity under conditions of higher folate nutrition status. The authors proposed that the folate itself could protect MTHFR activity by stabilizing the MTHFR protein and therefore allowing it to function normally. And I've given you the URL for this study that speaks to this. I think it's also interesting that the National Institute of Health genetic home reference, I've given you the URL at the bottom for the PubMed publication, that folic acid Number one, it has a robust safety record. It's safe, non-toxic doses. Higher levels are more likely to support good health if a person has a genetic issue, where the MTHFR enzyme is weaker than usual. It has been shown in many studies that because of the extra folic acid, because it converts to 5-MTHF, it's the precursor to MTHFR, it creates an appropriate large pool of the enzyme to allow for proper detoxification of homocysteine, even with weaker enzymes being produced. Just as an FYI, B2's importance in the MTHFR gene mutation. Folate has an important metabolic interaction with riboflavin, with B2. Riboflavin, or B2, is a precursor of the coenzyme flavin, adenine, dinucleotide, and certainly often short-formed as FAD or FAD. FAD is required for the activity of the folate metabolizing enzyme, MTHFR. So MTHFR is FAD dependent and in catalyzing and in converting THF into MTHF, or the active form of folate. Along with other B vitamins, higher riboflavin intakes have been associated with decreased plasma homocysteine concentrations as well. And I think what's interesting to note is that the riboflavin on folate metabolism appears to be greatest in individuals who have the polymorphism or of the 677CT, which I spoke about. Now, elevated, if we go to the last bullet, elevated homocysteine concentrations in these individuals, it's highly responsive in lowering the homocysteine levels when they're supplementing with B2, confirming the importance of riboflavin MTHFR interaction. So it's pretty interesting. The folate pathway, again, showing the methionine synthesis using folate, how folate is used in the methylation reaction. Nutrients supporting methylation, the enzymes and the pathways, folic acid, B12, B vitamins, selenium, methionine, TMG, DMG, N-acetylcysteine, and TMG and DMG, trimethylglycine or dimethylglycine, taurine. Speaking a little bit now, just briefly, about EGCGs. There were studies, and I've given you the URL for an article that was written, the reversal of hypermethylation, too much methylation, and the reactivation of genes by dietary polyphenic compounds. It's pretty remarkable because we know what we can do for hypomethylation, certainly the B12, the folic acid, etc. Hypermethylation has been shown polyphenic compounds will slow down the overactivity of methylation. Magnesium and molybdenum. Giving you a brief outline of what your diet might look like if you want to raise methylation activity in the body. Multivitamin, absolutely. A multivitamin and mineral. The asterisk indicates when you may take these supplements. SAMI, 200 milligrams with B6 and folic acid. You would take it breakfast, lunch, and dinner. TMG, or it might be DMG, but TMG is 1,000 milligrams. TMG is a form of a B vitamin, although and this is saying very loosely, so it is water soluble, and if you take more than what the body needs, then you will simply eliminate it. But 1,000 milligrams of TMG, and you would take this at breakfast. That's one example. It could be with food at any time. Methylcobalamin is away from food, and again the methyl form of B12. Magnesium with food, but away from the multivitamin. A B complex with food but away from the multivitamin. N-acetylcysteine with food. Our N-acetylcysteine at 600 milligrams also contains selenium. DHA, vitamin C and E, and probiotics all recommended as well. The now supplements that are available to you that would be complementary for this particular presentation and for methylation are listed here. We have an array of different types of SAMe. We certainly are notorious for our regimen of amino acids and DMG and TMG as well, magnesium, B vitamins. Thank you very much for joining me today. Have a wonderful day. Bye now.